Assalamu alaikum. Good morning. Today we'll talk about the transport of respiratory gases in the blood. And we mean by the respiratory gases oxygen, transport from the lungs to the tissues, and carbon dioxide transport from the peripheral tissues back to the lung. We'll see the forms of transport and different factors that affect the transport of gases in the blood. The objectives of the current lecture are to explain how the blood transports oxygen, draw an oxygen hemoglobin dissociation curve showing the relationship between oxygen partial pressure, hemoglobin saturation, and blood oxygen content. To understand the factors that affect the affinity of hemoglobin for oxygen, at least 5 different factors affect the affinity of hemoglobin for oxygen, and to differentiate between fetal and maternal hemoglobin in terms of their molecular structure and their affinity to oxygen. And list the forms by which carbon dioxide is carried in the blood. Now, the transport or regarding the transport of oxygen in the blood, each 100 ml of oxygenated blood that is pumped from the heart into the systemic circulation contains the equivalent of 20 milliliter of gaseous oxygen. This 20 milliliter is carried either or transported either in the form of dissolved oxygen in the plasma, and this is only 1.5% of the total oxygen transport, and it represents 0.3 ml out of the 20 ml of gaseous oxygen. The majority of oxygen, which is 98.5% or 19.7 milliliters of oxygen that is transported in the blood, is bound to hemoglobin inside the red blood cells. The heme portion of hemoglobin molecule contains 4 iron atoms, and each iron atom can bind 1 oxygen molecule. So in this equation here, we see that reduced hemoglobin or deoxyhemoglobin binds to oxygen in the lungs to form oxyhemoglobin that will circulate in the blood to reach the peripheral tissue, and in the peripheral tissue oxygen will dissociate from oxyhemoglobin. It will be released to the cells to be used in aerobic metabolism, and this will lead to the reformation of reduced hemoglobin or the oxyhemoglobin. Oxygen bound to hemoglobin, which is 98.5%, is trapped inside the red blood cells. So only the dissolved oxygen, which is 1.5% of the total transported oxygen, can freely diffuse out of the tissue capillaries in the tissue cells. Understanding the factors that determine binding and dissociation of oxygen with hemoglobin is very important, because the red blood cells in the peripheral tissue have to release that oxygen which is bound to hemoglobin to the plasma, and then from the plasma to the peripheral tissue cells. Here we can see a hemoglobin molecule. An adult hemoglobin molecule is composed of 2 alpha chains and 2 beta chains. So each hemoglobin molecule is composed of 4 polypeptide chains, and the adult hemoglobin is composed of 2 alpha and 2 beta. In the center of each polypeptide chain there is one heme ring that contains iron, and oxygen binds to hemoglobin molecule through this iron that is present in the center of the heme ring. Since we have 4 heme rings in one hemoglobin molecule, then one hemoglobin molecule can carry 4 oxygen molecules. So when hemoglobin molecule is fully saturated with oxygen, it means that that hemoglobin molecule is carrying 4 oxygen. What are the factors that affect the affinity of hemoglobin to oxygen? What we need is to have high affinity of hemoglobin for oxygen in the lungs, so it can carry the maximum amount of oxygen when it is in the lung, and we want to decrease the affinity of hemoglobin for oxygen in the peripheral tissues. So hemoglobin can release the highest amount of oxygen to the tissue because that oxygen is used in the aerobic metabolism. So the factors, number 1, the partial pressure of oxygen in the blood. And this is the most important factor that determines the affinity of hemoglobin for oxygen. Number 2, the acidity of the blood or the peripheral tissue or the pH. Number 3, the partial pressure of carbon dioxide. Number 4, the temperature. And the 5th and last factor is 2,3-bisphosphoglycerate. It is a metabolic product of the glycolysis that happens in the red blood cells, and we'll see how it will affect the affinity of hemoglobin for oxygen. Here we see a very important curve. This is called the oxygen hemoglobin dissociation curve. The y-axis represents the partial pressure of oxygen in the blood. The x-axis represents the partial pressure of oxygen in the blood, while the y-axis represents the percent saturation of hemoglobin. So as we go up, it means hemoglobin becomes more and more saturated with oxygen. And as we go to the right on the x-axis, it means we have higher partial pressure of oxygen in the blood. So the highest partial pressure of oxygen in the blood is present in the oxygenated blood, which is 100 millimeter mercury. The lowest is present in the veins that carry blood back to the heart, which is the deoxygenated blood in the systemic veins. We can see much lower partial pressure of oxygen in the blood in actively contracting muscles that extract large amount of oxygen, so the partial pressure of oxygen can go down to 20 millimeter mercury. So as we go to the right here up with the curves, it means that the hemoglobin affinity for oxygen is becoming higher and higher since it contains more oxygen, or it carries more oxygen. So as we go up to the right, it means higher affinity. As we go down, it means we have unloading of oxygen from hemoglobin. So in the veins or in the tissue where the partial pressure of oxygen drops and the cells down to 40, hemoglobin affinity for oxygen will decrease because the partial pressure will drop, so hemoglobin will release more oxygen to the cells of the peripheral tissue. The partial pressure of oxygen is the main factor that determines the percent hemoglobin saturation or the hemoglobin affinity for oxygen. At high partial pressure of oxygen, and this is the case in the lungs, more oxygen binds to hemoglobin, as I explained on the curve in the previous slide. So here in the lung we have oxygen, hemoglobin saturated 100% with oxygen. So more oxygen binds to hemoglobin, the reduced hemoglobin is converted to oxyhemoglobin, hemoglobin becomes fully saturated, which means each molecule carries 4 oxygen molecules. At low partial pressure of oxygen, and this is the case in the tissues when the partial pressure drops to 40 millimeter mercury, oxygen dissociates from hemoglobin, oxyhemoglobin is converted to reduced hemoglobin. Hemoglobin becomes partially saturated. In a normal person at rest, usually 1 out of the 4 oxygen molecules per 1 hemoglobin molecule will be released to the tissue. So the hemoglobin will not release all the 4 oxygen molecules that it carries, it will release only 1 molecule of oxygen to the tissue, and this is again at rest. The plateau part of the curve is where the partial pressure of oxygen is high, 100 millimeter mercury, and this is the case of the lung. The steep part of the curve exists at the systemic capillaries where hemoglobin unloads oxygen to the tissue cells. The other factors that affect affinity of hemoglobin to oxygen are acidity, partial pressure of carbon dioxide, temperature, and the concentration of bisphosphoglycerate inside the red blood. Let's see how acidity or pH affects the hemoglobin oxygen dissociation curve. Let's agree that any shift in the curve to the right means low affinity of hemoglobin to oxygen, and any shift of the curve to the left means that hemoglobin has higher affinity for oxygen. So here we can see that at low pH, 7.2, which is less than the normal pH, the curve shifts to the right, which means that increasing acidity enhances the unloading or the release of oxygen from hemoglobin, because hemoglobin affinity for oxygen will decrease. So this will shift the curve to the right. Low acidity, which means high pH, will increase the affinity of hemoglobin for oxygen, so the curve will shift to the left. The partial pressure of carbon dioxide at high levels of partial pressure of carbon dioxide, this will cause a shift in the curve again to the right, as we can see here. So high levels of carbon dioxide will decrease the affinity of hemoglobin for oxygen and will shift the curve to the right. Low levels of carbon dioxide will increase the affinity of hemoglobin to oxygen and will shift the curve to the left. The effects of pH and partial pressure of carbon dioxide on the hemoglobin oxygen dissociation curve is called the Bohr effect. So high hydrogen or low pH, high carbon dioxide will decrease the affinity of hemoglobin for oxygen, and this is called the Bohr's effect. The effect of temperature on the oxygen hemoglobin dissociation curve within a limit, within the range that is compatible with life. As temperature increases, more oxygen is released from hemoglobin, and this will shift the curve to the right. At low temperatures or at low body temperature, which is called hypothermia, more oxygen remains bound to hemoglobin because the affinity of hemoglobin to oxygen will increase, and this will shift the curve to the left. The last factor that affects the affinity of hemoglobin to oxygen is the concentration of the bisphosphoglycerate. Bisphosphoglycerate is formed by red blood cells during glycolysis. When it binds with the hemoglobin, it will decrease the affinity of hemoglobin to oxygen, so this will help in unloading oxygen from the hemoglobin molecule. Now we have different forms of hemoglobin, and the percentage of each form is different at different age groups. Let's compare the fetal and maternal hemoglobin. The maternal hemoglobin is mainly hemoglobin A, which is composed of 2 alpha chains and 2 beta chains, as I explained in the previous slides. Fetal hemoglobin, on the other hand, the beta chains are replaced by gamma chains, so the fetal hemoglobin or hemoglobin F is alpha 2 gamma 2, and it is different from the adult hemoglobin or hemoglobin A, which is alpha 2 beta 2. They differ in structure, and also they differ in their affinity for oxygen. Hemoglobin F has higher affinity for oxygen than hemoglobin A, which is the maternal hemoglobin, because hemoglobin F binds with the bisphosphoglycerate to a much lesser extent. So if less bisphosphoglycerate binds to hemoglobin F, it means that hemoglobin F will have higher affinity to oxygen compared to hemoglobin A. So this difference in the affinity of hemoglobin A between hemoglobin A in the maternal blood and hemoglobin F in the fetal blood will cause or will make the maternal hemoglobin oxygen dissociation curve a bit shifted to the right compared to the fetal hemoglobin. Hemoglobin F can carry up to 30% more oxygen compared to hemoglobin A. As maternal blood enters the placenta where the partial pressure of oxygen is very low, oxygen can easily be transferred to the fetal blood, and this is helped by the high affinity of hemoglobin F to oxygen. Now let's talk about the transport of carbon dioxide in the blood. And this transport happens between the peripheral tissues that produce carbon dioxide during metabolism to the lungs, where we get rid of carbon dioxide through the gas exchange to the alveoli, and then to outside the body by exhalation. Carbon dioxide is transported in our blood in three main forms: either as dissolved carbon dioxide in the plasma, and this accounts for 7% of carbon dioxide transport, and when this dissolved carbon dioxide in the plasma reaches the lungs, it diffuses in the alveoli or in the alveolar air, and it will be exhaled. The second form of carbon dioxide transport is bound to hemoglobin inside the red blood cells, and this represents about 23% of carbon dioxide transport in the blood. It binds to amino acids either in the alpha or beta chains of hemoglobin molecule. So carbon dioxide doesn't bind to iron in the heme ring, so it doesn't compete with oxygen on hemoglobin. So carbon dioxide and oxygen, they bind to different sites on hemoglobin, so there is no competition between these two respiratory gases. As we can see in this equation, hemoglobin here, when it binds with carbon dioxide, and this binding occurs in the peripheral tissues or in the systemic capillaries of the peripheral tissues, will produce carb-amino hemoglobin. Carb-amino, which means carbon dioxide bound to the amino acid of hemoglobin molecule. In the lungs, carbon dioxide will dissociate from hemoglobin to release free carbon dioxide that will be released from the red blood cell into the plasma. This carbon dioxide will cross the respiratory membrane and leaves the blood to the alveolus. This reaction is influenced by the partial pressure of carbon dioxide. At the pulmonary capillaries, the partial pressure of carbon dioxide is low, so carbon dioxide will dissociate from hemoglobin, while in the tissues, the partial pressure of carbon dioxide is very high due to the metabolic processes, and this will promote the formation of carb-amino hemoglobin molecules. The third and the main form of carbon dioxide transport is the transport of carbon dioxide as bicarbonate ions in the plasma. But to convert carbon dioxide to bicarbonate, we need an enzyme that is present in the red blood cells. So the carbon dioxide that is released from the tissues to the systemic capillaries. 70% of it will be converted inside the red blood cells to bicarbonate, and then it will be released into the plasma there in the systemic capillaries, and it will be carried back to the lung in the plasma as bicarbonate. And the process will be reversed in the lungs, as we'll see in the coming slides. So the transport of carbon dioxide as bicarbonate, carbon dioxide diffuses into the red blood cells. And inside the red blood cells, it combines with water to form carbonic acid. And this reaction needs the presence of the enzyme carbonic anhydrase. Carbonic acid, after its formation, will quickly dissociate into hydrogen ions and bicarbonate ions. As we can see here in this equation, carbon dioxide and water will form a carbonic acid, and this step is catalyzed by carbonic anhydrase enzyme. Then carbonic acid will quickly dissociate into hydrogen and bicarbonate ions that will leave the red blood cells and enter the plasma. In red blood cells, carbonic anhydrase reversibly catalyzes the conversion of carbon dioxide and water to carbonic acid. Let's have a closer look how carbon dioxide is transported from the tissues to the lung. Here we have at the tissue level, we have cells that continuously produce carbon dioxide, so its level in the cells is high. Down the partial pressure gradient, carbon dioxide will cross the interstitial fluid, the capillary membrane, and enter into the plasma. 7% will dissolve in the plasma and will be carried to the lungs as dissolved carbon dioxide. 23% will enter the red blood cells, bind to hemoglobin, and will form carbaminohemoglobin. The majority, which is 70%, will enter the red blood cell, will combine to water in the presence of carbonic anhydrase to produce carbonic acid that will dissociate into bicarbonate and hydrogen. Bicarbonate in the systemic capillaries will leave the red blood cells in exchange for chloride, and this exchange of a negatively charged ion with another negatively charged ion will maintain the electrical stability of the red blood cell, and this exchange is called the chloride shift. To counterbalance the outrush of negative bicarbonate ions from the red blood cells, chloride ions will move from the plasma into the erythrocytes, and this chloride shift will be reversed in the lungs, as we'll see in the coming slide. At the lung level, the opposite will happen. The dissolved carbon dioxide, which is 7%, will leave the plasma, will cross the respiratory membrane to enter into the alveolus down the concentration gradient. The 23% of carbon dioxide that is carried as carbaminohemoglobin will dissociate from hemoglobin, will enter the plasma, cross the respiratory membrane to enter into the alveolus. Now the bicarbonate, or the 70% of carbon dioxide that is carried as bicarbonate in the plasma, will enter the red blood cells in exchange for chloride in the process of chloride shift to maintain the electrical stability of the cell. The bicarbonate inside the red blood cell will combine with hydrogen to form carbonic acid that will dissociate in presence of carbonic anhydrase to water and carbon dioxide. Carbon dioxide will leave the red blood cell to the plasma, will cross the respiratory membrane, and will enter into the alveolus. Again, to counterbalance the shift of negative bicarbonate ions into the red blood cells, chloride ions will move from the erythrocyte to the plasma in the process of chloride shift. Here in this slide we see a summary of the ways of transport of oxygen and carbon dioxide between the lungs and the peripheral tissue. As I explained, in the lungs. The blood will be oxygenated, and 98.5% of oxygen will be carried as bound to hemoglobin as oxyhemoglobin, and only 1.5% will be dissolved in the plasma. The oxygen will be carried to the tissue, and then dissolved oxygen will move or leave the plasma to the cells, and the oxygen bound to hemoglobin will dissociate from hemoglobin and will leave the blood to the cells, and the carbon dioxide that is a product of metabolism will be carried in the three different forms as bicarbonate, carbaminohemoglobin, or dissolved carbon dioxide in the blood back to the lungs, where carbon dioxide will cross the respiratory membrane to enter into the alveolus. There is something called the Haldane effect. The Haldane effect, as we saw in the factors that affect the affinity of hemoglobin for oxygen, and one of these factors is the concentration of carbon dioxide. Also, the concentration of oxygen in the form of oxyhemoglobin can affect the carbon dioxide carrying capacity of hemoglobin. So the amount of carbon dioxide bound to hemoglobin is affected by the percent saturation of hemoglobin with oxygen. So if we have high amount of oxyhemoglobin inside the red blood cells, this means that this hemoglobin can carry less carbon dioxide. If we have less amount of oxyhemoglobin, it means that the affinity of this hemoglobin will be higher for carbon dioxide, and hemoglobin can carry more carbon dioxide. So the lower the amount of oxyhemoglobin in the blood, the higher the carbon dioxide carrying capacity of hemoglobin. This is called the Haldane effect. The effect of carbon dioxide on the affinity of hemoglobin to oxygen is called the Bohr effect, while the effect of oxyhemoglobin on the carbon dioxide carrying capacity is called the Haldane effect. Okay, so in conclusion, hemoglobin promotes the net transfer of oxygen at both the alveolar and tissue levels. Why? Because hemoglobin carries 98.5% of total oxygen that is transported in the blood. There is a net diffusion of oxygen from the alveoli to the blood. This occurs continuously until hemoglobin is as saturated as possible. At the tissue level or at the peripheral cells, hemoglobin rapidly delivers oxygen into the blood plasma and then to the tissue cells, and we studied the various factors that promote this unloading or this dissociation of oxygen from hemoglobin. One of them is carbon dioxide through the Bohr's effect. An increase in carbon dioxide in the tissue cells, and this is usual due to the continuous production of carbon dioxide during metabolism, and its release into the systemic capillary will increase hemoglobin dissociation from oxygen, and as we saw previously, this will shift the hemoglobin oxygen dissociation curve to the right. High metabolism will cause high acidity or low pH. So increased acidity will shift the hemoglobin oxygen dissociation curve to the right, which means more dissociation of oxygen from hemoglobin in the systemic capillaries, or more unloading of oxygen to the tissue cells. Another factor is the temperature, and we know that metabolically active tissues they generate high temperatures. So high temperature also decreases the affinity of hemoglobin to oxygen, and this will shift the curve the hemoglobin oxygen dissociation curve to the right, and the same also is the high concentration of bisphosphoglycerate inside the red blood cells. This shift of the curve to the right, which means more dissociation of oxygen from hemoglobin, is called the Bohr's effect. There is one thing about hemoglobin which is very special: its affinity to carbon monoxide. When fuel or carbon is incompletely burned or inside closed, poorly ventilated room, instead of production of carbon dioxide, large amount of carbon monoxide will be formed, and this can lead to carbon monoxide poisoning and even death. Why this happens? Because carbon monoxide doesn't bind to the amino acids of the polypeptide chains of the hemoglobin molecule. It competes with oxygen for the iron in the heme ring. And the problem with carbon monoxide that it has 200 times more affinity for that iron on the heme ring compared to oxygen. So if carbon monoxide is present in large amount in the environment of a room, then hemoglobin is no longer able to carry any oxygen from the lungs to the tissues, and this can lead to death. And this is the main problem of carbon monoxide poisoning. And the binding of carbon monoxide to hemoglobin is called carboxyhemoglobin. Please be careful. Many students are confused or mix between carbaminohemoglobin and carboxyhemoglobin. Carbaminohemoglobin means carbon dioxide carried on hemoglobin bound to the amino acids of the polypeptide chain of hemoglobin. Carboxyhemoglobin is carbon monoxide bound to hemoglobin with high affinity at the iron that is present or attached to the iron that is present in the center of the heme ring in the polypeptide chains. Thank you very much for your patience. I welcome any email containing questions or inquiries about this lecture. Thank you, and meet you in the next lecture.